Aude is the MIT director of the MIT Watson AI Lab and the co-director of the MIT Quest for Intelligence and a senior research scientist at the Computer Science and AI Labs at MIT. She received her PhD from the Institut National Polytechnique of Grenoble, France. Her research is truly cross-disciplinary, spanning human perception, cognition, computer vision, and cognitive science and neuroscience. And she has made seminal contributions in all of these fields over the years. Her talk titled Resolving Human Brain Responses in Space and Time. Welcome, Aude.
Thank you very much, Ratan, for this introduction. Hello, everyone. It's a pleasure to give you a tour of some of the neuroscience work we have been doing in my lab for the past few years on resolving human brain responses in space and time. So I do have a fascination for complex systems, and the human brain is certainly one of the challenger systems to try to understand. And there's a simple fact about the human brain as a neural network. It's a very dynamic neural network. Information is processed in time. When you see, hear, or touch something, the response starts in the sensory cortex and then move forward, move and spread to other regions. So we do not know very well how to deal with the timing and sequential processing of information or when to integrate different source information in artificial neural network. So if we want to understand both human intelligence and see how at some point we will be able to interact well with artificial systems that are going to help us memorize or even teach us things or during this interaction, I believe that we need more fundamental knowledge about how the human brain processes information over time. So today I will present 3 results related to the spatial temporal map of human brain responses and ways to try to capture the timing of processes. And so in this research program, we propose to build what we call spatial temporal maps of human brain processes for different modalities and different tasks. So what does it mean? So basic sensory processing is you are listening or touching a surface or looking at something. So can we have a full brain picture of when some regions are activated and what they process and where the information goes next? Recognition and memory, when you put an information into memory, where does it go? Is it going to last? How long does it take for that new image or that event or that things you hear to reach memory system? Learning plasticity, also multisensory integration. Our brain is always integrating information for vision and sound and touch and so on. So there's a lot of work in neuroscience and behavioral science about when it is the right time to integrate information across sensory modalities, but there's actually not much work showing when some part of the brain do integrate vision with audition and so on. Working memory, attention, it involves a lot of the prefrontal and frontal cortices. So when you rehearse something or try to put something into memory, there's a lot of timing information going there. There's also a lot of hope for spatial temporal maps in clinical research. If you have some tasks that are disrupted in people with certain disease like autism or schizophrenia or other aspect that disturb the whole global processing of information in the brain, there's a lot of insight that could be gathered if we can see how a healthy brain or a brain with certain disease process information over time. And also for anything related to brain-machine interface, deep neural network comparison across species, having the timing is important. So it's still understanding the timing of processes, an understudied dimension in cognitive computational neuroscience itself. So today I will present 3 results one about basic sensory processing of vision, one about audition. We have a new bioarchive paper, and new results that I have not presented yet to anyone about the spatial temporal map of visual memory. So capturing both time and space in the human brain in a non-invasive way, there are not a lot of tools to do that yet. It's almost, we are at the cavern age of cognitive neuroscience in that domain. So in a couple of decades, there might be some measurement and devices that will allow to have a better grasp. But right now we can do quite a bit of things with what we have. And I'm very lucky at MIT to have access to two types of machine that capture different type of information measuring the brain. And I will show you today how we can gather some insight for some of the basic tasks using those two machine. So one is MEG or magnetoencephalography. Let me say you can also use EEG for this. So MEG is a functional neuroimaging technique for mapping brain activity by recording magnetic field produced by the electrical current that naturally occur in the brain when you see, hear, or do something. And this is using very sensitive magnetometers. So you have an example here on the left. And then that will capture every millisecond responses in the brain. So now regarding space or where there's responses in the brain, you can use fMRI, functional magnetic resonance imaging, that measure the brain activity by detecting changes associated with blood flow. And so this technique relies on the fact that the cerebral blood flow and neuronal activation are coupled. So when an area of the brain is more in use, then we can have a response. So in a collection of work that we started in 2014 with Radosław Ciszy, we combine the data from the MEG and from the fMRI using representational geometry, RSA analysis from Nico Kriske-Gorte, and a large collection of machine learning method. So let me give you a quick tour of the method and then some of the results. So we put our subject in both the MEG and the fMRI scanner. So let's start with MEG. So we show, for instance, collection of image, and we can gather responses every time we show an image, and we repeat them and so on. And let's say we show 100 image. We calculate those matrices of dissimilarity by looking at the pairwise comparison between every two of our stimuli. And there's different method to do the pairwise comparison and to calculate those distances matrices. And there's a lot of very nice work out there that can tell you what's the best distance matrix to use in order to build your matrices. So we can do that for MEG while the subject look at images. And what do we get? Well, we get those similarity matrices that tell us how close the signal of two images, for instance, is here, the hand and the faces are together. So only with the MEG, we can actually see already a lot of structure that emerge over time. So we are still negative time and now positive time around 60, 70 milliseconds. You can see in the MDS representation shown here that after 120 milliseconds, there is some coherent signal that put together various images here of faces, body, or inanimate object. Let me run it again. See again the responses. All right. I find that pretty amazing that so much structure can be captured with MEG, that it's always a pleasure to show those movies. All right, so there's a lot of structure and signal into the MEG on this information. So now what about the fMRI? So we can do a similar analysis. First, we put our subject into the fMRI. We can run the same experiment showing images to our subject in the scanner. And we can look at a little region in the brain using searchlight, big region, small region, the whole brain or only certain region of interest, and again look at the distances or pairwise stimuli within that space and build matrices of similarity or dissimilarity for different brain regions. And the method we propose with Wadoslo Chishi was then, well, you have matrices that organize stimuli in a certain way, and we can look at how close those matrices are in MEG time or fMRI voxel, for instance, by using a Spearman correlation. And by looking at the correlation that are above statistic threshold, it basically gives you a mapping of when information in time has a higher correlation with some voxel or region in the human brain. And after showing plenty of images to subject, here we put together 15 subject brain. We can compute what we call the spatial temporal maps that show the highest correlation between MEG and fMRI for here visual image and recognition of object. So that gives us the most significant higher correlation in that space. There's a lot of information we are missing. There's a lot of things happening that this method cannot capture or show us. However, it's like having a sight of the top of the iceberg of all the information that can be captured. And as more better techniques arise and better devices are being built in order to have a better sense of the timing in the human brain, you could see how we could gather pretty nice information about the other regions that are also activated. So if I run it again, so this was for vision, and we can see some region in the occipital area that are really highly activated. When you look at image, there is no mystery in this spatial temporal map I show you. And what we can do then, and we have many papers over the years, is we can go and carve and can look at a particular region and look more at the representation of the MDS into that region, how close some stimuli are, the cluster we see, and so on. So even if I show you here a movie of the whole brain, there's then all the work to go and carve more specifically what some regions are doing and when. And if you are interested in this method, we have a few papers with Chishi over the years, and also a review in 2020. And here I'll show you a snapshot of two of our visual data set. So we spent a few years replicating those results, as well as using different type of data set and hypotheses. So basically, everything you will find on this method so far from our group was done on vision, image, visual recognition. But it was also very important to go really step by step. And when we were observing a region that was reacting at a certain time, it was very important to double check if this was found by other people. So basically we could show to which extent the method is valid with a result that we know are very classical in how visual information is processed in the primate brain. And all the data related to the three data sets that we use in vision, here I show two data sets, they are all MEG and fMRI, all the subject data are available on our website, so you can go and pick up, gather the data, do your own analysis, and find the same or find something new or find something different. There are many ways to really cut that piece of data. So in visual perception, using those spatial temporal map, our goal was really to check our method. So a lot of the results we found were found by 2 or 3 decades of work by many other groups. So that was really very important to show, so the hierarchy of response. And the spatial temporal maps and the region that we found align with many previous results. One result that we found that was new, and it's published in the paper shown at the bottom, is for recognizing object, both ventral and dorsal region responses around the same time. It's not that the ventral was responding earlier than the dorsal. So there are out there some results that are similar to this, but seeing really the timing and the spread of information in those two different regions at the same time was something that was pretty exciting and elegant to see in this work. And there may be many lessons to learn in order to build artificial neural network that are closer to the human brain, but certainly one of them out of this line of work that can be inspiring for model is, it looks like certainly for visual recognition, there are different high-level representation for objects after an initial command processing. It's almost like the initial stage, like the visual cortex, the process information. And then there's this separation that is very well known in neuroscience between ventral and dorsal. This is something that the brain is implemented for vision, and it did not implement that for audition. How do we know? Because we play the same game of the spatial temporal maps now for auditory stimuli. So in more recent work, we took a new set of subjects, and now we had the subject both in the MEG and the fMRI listening to sound, snippet of sound of half a second each. So half a second is both very short, but it's actually largely enough to know what the type of sound, very clearly the category of sound you're listening to. So we had people listening to 80 sound, a different voice, accent, different people, as well as different objects like the telephone, the door opening, different sounds from environment like the wave, the storm, orchestra, traffic jam, as well as sound from animals. So there were those various categories and all mixed up. So you're in the scanner and you're listening to those sound and you do an orthogonal task. And we can again combine the signal both from MEG and fMRI and look now at the spatial temporal maps when you're listening to sound. And here it is. So sound, left, right, information is going to start near your ears. Indeed, that's absolutely what we see. But what was very, very interesting for us, and you're going to have a sight of this just looking at those images. We are talking about sound of half a second. And you can already see the quantity of brain and neural matters that is involved into processing the sound. Just as a first sight, it's like it feels that it's much more than vision. And again, those are the correlation that exceed a certain statistical threshold, and we are pretty severe with our threshold. But you can see that over 500 milliseconds, there's a lot going on in the sound perception space. Let me play it again. So start around, let's see, 70, 80 millisecond signal, and it spread very rapidly to the temporal, and then some responses that you can see here in prefrontal and then higher up. And then I'm going to show you more results. The responses goes into the fusiform and the parahippocampal area and the lateral complex. It's really an orchestra of brain responses to only snippet of sound. And it does show the richness and variability. And indeed, for the brain, processing sound in terms of signal coming in, but then making meanings out of it is actually involving a lot more of cortical areas. So it will take us years to go and deep down analyze all those results. Let me give you some more snapshots of what we found for sound perception. So what we are interested in is, again, the timing. And here I show you the peak latency of responses in milliseconds of a collection of region of interest. So out of the movie you just showed, we can have the entire brain or just go selectively look at a particular region in the brain. And the early auditory region, primary auditory cortex PAC, TE12, planum temporal, and so on. They are, oh, I have a misalignment of my color here. Anyway, so there's around 115 milliseconds. You have the early auditory region that are responding to the sound. But then we see in the temporal voice area and the left inferior frontal gyrus around 200 milliseconds, the papers show specific signature for human voices and a very nice separation between human voices, animal, and the other sound. So into the processing of sound, there is those specialized area for human voices as well as animal sounds that are really coming up early on into to discriminate that those sounds are animate. And then we see a sort of further phase in regions that have been classically associated with vision, like the fusiform face area, parahippocampal place area, lateral occipital complex, that shows peak of responses to sound only around 300 milliseconds. So I refer to you to the bioRxiv papers, which have much more results regarding the timing and the region and the type also of responses that are more perceptual or semantic, the classes of stimuli that seems to be preferentially processed by those regions that I show here. So auditory perception, there's a lot of excellent work out there that gave information on the timing of sound perception. This work show for the first time the timing of both early responses to the sound in the auditory cortex, as well as semantic segregation and recognition in higher of a region. So it gives for the first time a view of sound in the entire brain. We did know that there were some regions specific for the human voice, and with some indication of the timing, now we see those two coming up around 200 milliseconds in the prefrontal that are really specific. So it's nice to have this timestamp. And then this late response around 300 milliseconds for auditory stimuli in region classically associated with vision. So there's going to be a lot more work that can be done on those data. In some of the preliminary analysis we do, there's more than 20 regions really strongly participating to the recognition of sound, and I'm only shown about 10 right now, so there's many more to put into this architecture of sound recognition. And some of the lesson for those of you working on model is human voices have a specialized module, and they are separated early on before the semantic separation of our sound. And that audition is mixed with visual features at a later stage in the hierarchy. So if you're building model of auditory and visual perception, your model, your artificial neural networks, you may find other ways to either combine the features of the two in order to recognize what's going on. But the brain seems to have implemented something for audition that is different than vision, and certainly for us, it seems to work. Yeah, we can understand what's going on. So keep going on to those spatial temporal maps. As I showed you in the introduction, there's many tasks that can be done in the scanner in order then to analyze the data and capture those spatial temporal maps. So one of the topics that have been going on for a decade now in our group is the notion of memorability. And here I just show a snapshot of some of the work that we have done. The notion of memorability is that there is some event that could be actually mostly visual event, but that also happened with words, and there's other work by other group with actually sound and speech and so on that are more memorable than others. So if you see a certain image, all of us will remember them. And if you see other image, all of us will forget them. And we have out there quite a bit of work on what makes an image or a video memorable to all of us and how long it takes for it to fade away. And there's very nice work by Wilma Benbridge, who has a lot of other papers on this notion of visual and auditory memorability. But we had access to the technique, the spatial temporal map. So we had the unique opportunity to now put together space and time and look at, okay, what are the signal in the brain in terms of the region that can differentiate that some images are more memorable and some are less memorable, and also the timing. So here I see the selection of images we showed. So all those images, there are two groups, some memorable, some less memorable, high and low. And they were selected by pairs of image, but they are all shown at random to subject in the two scanners. People have no idea that those are related to different level of memory. They see those images for the first time, one after the other in the scanner. And we just record the response. So for subjects, it's just a visual experiment, an orthogonal task, they look at image. But what we do is then we play this game, this analysis, where we have a movie for high memorable image, a spatial temporal movie for low memorable image, and then we do the difference. And the difference shows statistically the region and the timing where memorable images are higher. And what I'm going to show you next is the difference movie. So when the brain tells us, at least we can capture that an image is more memorable and will be actually memorized. That's what we also check later. As you can see, the memorability signal is late. So we have one actually in the higher region where we did expect it, but very late, 300 milliseconds and so on. And we have not put yet the paper on bioRxiv, give us a few weeks. But I just want to show you here a snapshot of those regions that we capture that show higher memorability signal. This is not in the, that's not an early signal, it's a late signal. Basically, you see images are processed in the early visual cortex. And then there seems to be a signal that those are memorable around 300 milliseconds in the posterior temporal lobe. And we have then a sort of feedback signal back to the early visual cortex later on. So we do have to interpret and analyze those data, but those are very new data showing a strong signal of memorability. And then independently, we know that the memorable image have a higher chance to make their way into memory and be remembered than the forgettable one. They do fade away, and we do see that they do fade away after 300-400 milliseconds already in the posterior temporal lobe. Maybe they will never have a chance to go into the memory structure. So memorability, late responses for memorable image. And the lesson from model, it looks like memorability can be used as a measure of the utility of information, which is something early on is like, not all images will be treated equally by your neural network. Some will have a stronger memorability, a stronger signal than others. And maybe those are related to the utility of information. What you put into memory is in the things that maybe that are stronger to remember or more important. So human artificial cognition are two things that I think are like the two sides of the same coin. And it's really very exciting in my group that we study both sides. We have the human brain, and then we do a lot of work on the neural networks. And for all the new generation out there, I really recommend you read about neuroscience and try to think about both neuroscience and artificial intelligence and neural networks, because there's so much things that can be, so much that we can find inspiring studying the two. Thank you.